Hi friends, welcome back to Sharing Zoology. In our previous session, we learned about cleavage and blastulation. See, in this section, I am going to explain about the structure of blastocyst and implantation process. First one is structure of blastocyst. Then human blastula called as blastocyst. Human blastula. Human blastula stage is called as blastocyst and structure of this blastocyst. This is the structure of blastocyst. In humans, this blastocyst consists a outer cell mass, a outer layer of cell. This is the outer cell mass, a outer layer of cells or outer cell mass, and a group of inner cell mass. So this blastocyst consists outer and inner cell mass. Then this is the cavity of this blastocyst. This cavity is called blastocele. So outer layer, outer cell mass, then inner cell mass and blastocele. This outer cell mass called as trophoblast. This outer cell mass called as trophoblast or ectoderm. So here the outer cell mass called the trophoblast or ectoderm, and this group of inner cell mass. This inner cell mass called embryoblast. So this is the structure of human blastula. This human blastula contain outer cell mass called as trophoblast or trophectoderm, and these are a group of inner cell mass called as embryoblast. And here, this inner cell mass contains, these are inner cell mass. This inner cell mass contains stem cells. Inner cell mass contains stem cells. See, there are different types of stem cells. Stem cells are adult stem cells, embryonic stem cells. Any of these stem cells are embryonic stem cells. And these stem cells have different potency. Some of the stem cells are totipotent, some are pluripotent, some are multipotent, and maybe unipotent. Here, the stem cells of this inner cell mass are pluripotent. Stem cells of inner cell mass are pluripotent. Pluripotency means have the ability to develop all type of cell. These stem cells have the ability to become all the type of cell. So this pluripotent means the potency to develop all the type of cell except this trophoblast. These are pluripotent stem cells because these cells have the ability to become all the cell type except this trophoblast cell. These cells are not able to develop these trophoblast cells. But we know the early embryonic stages include, we know about zygote. The zygote undergo division to form 2-celled embryo, then 3-celled embryo, 4-celled embryo, 8-celled embryo. Before compaction, I taught about compaction in my previous video. Anyway, before compaction, that means in zygote stage, or 2-celled embryonic stage or up to 8-celled embryonic stage. Zygote or 2 to 8-celled embryonic stage. The blastomere, we know blastomeres means each cell formed as a result of cleavage are blastomere. The zygote or 2 to 8-celled embryonic stage, the blastomeres are totipotent. Blastomeres are totipotent. The totipotency means have the ability to develop all the type of cell including this trophoblast. It have the ability to develop all the cell type including this trophoblast. That condition is totipotency. In our embryonic life, zygote or 2-celled embryonic stage or up to 8-celled embryonic stage, the blastomeres are totipotent. Then after compaction, they reduce that totipotency. Then here, these cells of this inner cell mass are pluripotent, have the ability to develop all the cell type except this trophoblast. So this is the difference between totipotency and pluripotency. Anyway, here these inner cell mass are pluripotent stem cells. So only this inner cell mass contain the pluripotent stem cells. So here these cells differentiated to form different types of cell, different types of tissues and different types of organ system and organism. So in future, during the development, suppose this is the trophoblast and this form this inner cell mass. In future, during the development, only this inner cell mass developed as the actual embryo. Only this inner cell mass developed as embryo. Because only inner cell mass contains stem cells for the development of different types of cells, organs and tissues. So the fate of inner cell mass is the inner cell mass is the embryo proper. The inner cell mass becomes the actual embryo. So the inner cell mass is called as embryoblast. This is why this inner cell mass called as embryoblast. Okay. And this outer cell mass is called as trophoblast or trophectoderm. We know fertilization takes place in this fallopian tube. After fertilization, the zygote undergo cleavage to form 2-celled embryo, 4-celled embryo, 8-celled embryo, then 16-celled embryonic stage is called as morula. This morula is converted to blastula. Human blastula is called as blastocyst. In this blastocyst, these are a group of inner cell mass and these are outer cell mass. This outer cell mass is called as trophoblast or trophectoderm. Here, trophoblast means the word trophin means to nourish. So the most important function of this trophoblast is drawing food for this embryo. The main function of this trophoblast is to nourish this embryo. Because before implantation or before the formation of placenta, this endometrium secretes some uterine milk. And that nutritive milk is absorbed and transported to this inner cell mass or this embryonic cell by this trophoblast. So the main function of this trophoblast is drawing food for the embryo. So functions of trophoblast. First function, this trophoblast is to nourish this embryo. Drawing food for the embryo is the main function of trophoblast. Second one, see here this trophoblast. This trophoblast get attached to the endometrium of uterus. The cells of this trophoblast get attached to the endometrium of uterus and helps in implantation. So another function, it helps in implantation. So drawing food for the embryo. Next one, it helps in implantation. Then after implantation, this trophoblast helps in the formation of placenta. And this trophoblast becomes a extra embryonic membrane. Extra means outside. Extra embryonic membrane means the membrane seen outside the embryo. See, here the inner cell mass become embryo. So here this trophoblast become the membrane seen outside the embryo. That is extra embryonic membrane called as chorion. This trophoblast becomes the extra embryonic membrane chorion. So these are the functions of trophoblast. So functions of trophoblast are trophoblast main function drawing food for the embryo. Then it helps in implantation. Then it becomes extra embryonic membrane chorion and helps in the formation of placenta. This forms the structure of blastocyst. And this blastocyst reaches the uterus and get implanted. Next is implantation. Implantation means the blastocysts get attached to the endometrium of uterus. So after fertilization, within the uterus, the blastocyst stage of embryo get attached to the endometrium of uterus is called as implantation. The blastocysts get attached to the endometrium, and as a result, this blastocysts get buried in endometrium. That process is implantation. See, let's see the enlarged view of endometrium. We know the endometrium have two layers. This is the basal layer of endometrium and this one is the superficial layer of endometrium. This basal layer of endometrium is called as stratum basalis or decidua basalis. Stratum basalis or decidua basalis. And this is the inner layer or the superficial layer. This layer is called as the decidua functionalis. Due to the deficiency of progesterone, this decidua functionalis slough off and released from the uterus during menstruation. Anyway, this is the decidua functionalis and this one is decidua basalis. These are the two layers of this endometrium of uterus. Then the extracellular matrix of this endometrium made up of fibronectin, laminin, endocrine, complex sugars, collagen, then heparin sulfate receptors, sulfated polysaccharides, etc. So the extracellular matrix of this endometrium epithelium is formed of fibronectin, laminin, endocrine, complex sugars are there, and collagen protein, heparin sulfate receptors, some sulfated polysaccharide, etc. are found in the extracellular matrix of this endometrium epithelium. Then this is blastocyst. We know the trophoblast of this blastocyst first get attached to the endometrium for implantation. One of the functions of trophoblast is helps in implantation. For implantation, the L-selectin is a transmembrane glycoprotein. is a transmembrane glycoprotein on the trophoblast. So first of all, the L-selectin. What is L-selectin? Is a transmembrane glycoprotein on this trophoblast get binded to the sulfated polysaccharide of this endometrium. This is the initial binding. The L-selectin of trophoblast attached to the sulfated polysaccharide of this endometrium of uterus. After this initial binding, several adhesion proteins appeared here to coordinate the binding more effective. Then these trophoblast cells, these are trophoblast. These trophoblast cells synthesize integrins. This integrin is another transmembrane protein and it is synthesized by here in this trophoblast cell synthesize this integrin. This integrin then bind to the uterine laminins, fibronectin and collagen. So this integrin bind with the uterine laminin, fibronectin and collagen proteins. So the trophoblast cells secrete integrin and which helps to bind these type of extracellular matrix protein of this uterus. Then now this blastocyst is attached to the endometrium. And here this trophoblast as well as the endometrial cells produce cadherin, cell adhesion protein cadherin. And this cadherin also helps to fix this blastocyst to the endometrium. This cadherin also helps to fix this blastocyst to the endometrium. These are the extracellular matrix protein, polysaccharide, etc. are involved in this implantation. Then the cells of this trophoblast secrete. Cells of this trophoblast secrete some proteases enzyme. So cells of trophoblast secrete some proteases. That proteases digest this extracellular matrix. So that proteases digest this extracellular matrix. After the digestion of this extracellular matrix by the proteases secreted by this trophoblast, the uterine cells divide rapidly. The endometrial cells divide rapidly and cover this endometrial cell divide rapidly and cover this blastocyst. As a result, now this blastocyst is embedded in this endometrium. After this binding, the cells of the trophoblast split. So the cells of the trophoblast now split and form. See here now the cells of the trophoblast layer split. The trophoblast layer split to form two layers. Now this trophoblast split to form two layers. They are syncytiotrophoblast. Syncytiotrophoblast and this one is cytotrophoblast. Syncytiotrophoblast and cytotrophoblast. So the cells of the trophoblast split to form syncytiotrophoblast and cytotrophoblast. Syncytio means multinucleated condition. Syncytial condition, multinucleated condition. Here syncytial condition means here in this trophoblast cell, suppose these are the trophoblast cells formed by the splitting, and here these trophoblast cells disintegrate or loses their cell membrane. These cells disintegrate their cell membrane and all these membrane fused. What happened? Which resulted in the fusion of this cytoplasm and nucleus. Resulted the fusion of this protoplasm. What happened? It became a multinucleated condition. So in this syncytial trophoblast, the cell loses the cell membrane. After the disintegration of cell membrane resulted the fusion of protoplasm. That fusion of protoplasm causes a multinucleated condition. This multinucleated layer is known as syncytial trophoblast. And rest are known as cytotrophoblast. So syncytial trophoblast and cytotrophoblast. Here the cells of the syncytial trophoblast again secrete hydrolytic enzymes. Release hydrolytic enzymes. This hydrolytic enzyme helps to penetrate or dissolve the endometrium. What happened? As a result, this blastocyst completely penetrated and buried in this endometrium. So, when this syncytial trophoblast secrete hydrolytic enzyme which help to dissolve this endometrium, what happened? As a result, this blastocyst completely buried in endometrium. This process is called as implantation. Now this blastocyst is completely buried in endometrium. Simultaneous with this implantation, a number of changes occurs in the cells of inner cell mass. That means gastrulation takes place. I'll explain later about the gastrulation in my next video. And a number of changes occurs in this syncytial trophoblast and cytotrophoblast. And these will result in the formation of placenta. I'll explain later about this placenta formation also. So in this session we completed about the structure of blastocyst and the implantation. Okay, thank you for watching my video. Please like, share and subscribe my channel, Sharing Zoology.